So who can tell me what kinds of things can be infectious? Starting with the most complex. What kinds of things can be infectious? Eukaryotic pathogens. Okay, Bryant, can you give me any examples? Or can anybody give me examples of some eukaryotic pathogens? No. Okay, malaria is the disease. What's the organism? Okay, trypanosoma is one. And then which one causes malaria? Plasmodium, right. So those are the most complex things, eukaryotes. And of course, what's less than a eukaryote, but still a cell? Prokaryotes, right. So remember, we've talked about a lot of different kinds of prokaryotes. Salmonella, Shigella, Staph, Strep, all kinds of things. So what's no longer a cell, but is still infectious? Viruses, right. And today we are going to start talking about a lot of different animal viruses. And what's less than a virus? No, not yet. What's before prion? A viroid, right? So just a little naked piece of RNA, which is infectious. And then, of course, what's less than RNA are the proteins, like prion proteins. So it's just an interesting spectrum of things that can be infectious. But today we are going to start talking about animal viruses. I'm going to talk a little bit longer today because the lab is short. So remember, we talked about the stages of viral infection, and we said that all viruses follow the same 5 stages with some modifications, depending upon what kind of virus it is. But the first thing that any virus must do is attach to the host cell. And so animal viruses use cell surface receptors just like bacterial viruses do. But the animal viruses are attaching to a membrane, since we're talking about animal cells rather than a cell wall. And when it comes to penetration, here we have an important difference. Remember, the bacterial virus landed on the host cell, used lysozyme to drill a hole to the cell wall and inject the DNA, and the capsid and all the additional structures to stay on the outside of the cell. When it comes to animal virus, the entire virus moves inside of the host cell. So capsid and everything, the entire virus moves in, usually by receptor-mediated endocytosis, which we'll talk about as a normal cellular process. Cells use receptor-mediated endocytosis to move large quantities from the outside to the inside of the cell. And animal viruses have evolved to exploit that natural process. And so they land on the host cell, and the host cell thinks, well, this is something good that I want to swallow. I want to take it inside. And so the host cell actually infects itself. The cell can also, or the virus can also use penocytosis, another cellular process, or fusion, as in the case of membrane-bound viruses, where the membrane of the virus can fuse with the membrane of the host cell. But the entire virus moves inside. Once inside, the capsid will be broken apart to release the nucleic acid. And once again, that viral nucleic acid takes over. And the cell, the host cell, is forced to replicate it, to transcribe and translate it, to make new viruses. Now, at the release step, here we see another difference. There are some animal viruses those that are not surrounded by a membrane, that will lyse the host cells. So they'll destroy the host cells so they can be released. And so that's similar to bacteriophages. But the animal viruses that are surrounded by a membrane, here's where they acquire that membrane. They steal it from the host cell by budding. So the virus moves to the host cell membrane, and the membrane pinches off around the virus, and the virus buds and stealing host membrane. Now, eventually the host cell is going to die because it's losing membrane, cannot keep up with the membrane loss. So it will die, but not right away. And so it's possible, if the viruses bud slowly enough, that the host cell can live a significant period of time constantly producing new viruses. So, but this is an important difference between animal viruses and bacteriophages. Right? The bacteriophages rupture the cell. Here, a membrane-bound virus will bud from the cell, eventually killing it, but not right away. So here we see an example of fusion, where we have a membrane-bound virus, and the membrane can fuse with the host cell membrane, and that helps the virus to get inside. But for most animal viruses without membranes, they land on cell surface receptors, and the host cell engulfs them, surrounds them with membrane, forming a vesicle to move that virus to the interior of the cell. So animal cells actually do infect themselves, right? Because the viruses are tricking them, right? They're fooling those host cells into thinking the virus is something good to eat, something good to move inside the cell. And that's how most host cells get infected, is they actually infect themselves. When it comes to the release, where budding is the common term we use, the fancier term is exocytosis, right? The reverse of endocytosis. So the membrane-bound viruses, as they're being produced, they move to the cell membrane, and the membrane will pinch off around them and release those viruses. So we call that a bud. In this image here, we can see multiple buds all over the cell. As I mentioned, if the viruses bud slowly enough, that cell will survive for a while and potentially continue to produce many more viruses that way. When it comes to replication strategies, I used to talk a lot more about this, but the main idea is that any type of nucleic acid that exists, there's some virus that uses it. So we have DNA viruses, we have RNA viruses. They can be double-stranded DNA viruses or single-stranded DNA viruses, double-stranded RNA viruses or single-stranded RNA viruses. And when it comes to the directionality of the single-stranded viruses, that a DNA or RNA can be 5' to 3' or 3' to 5'. So basically, any type of nucleic acid that exists, there is some virus out there that uses it. And there's even a virus that is a parasite of other viruses. So these little viruses that cannot infect and force a cell to replicate them and make more copies of them unless that host cell is co-infected with another virus. And then the parasite uses the other virus's enzymes in order to make more copies of itself. So even among the viruses, there are viruses that parasitize other viruses. So viruses are very diverse. It's very amazing all the things they can do, especially when you consider how few genes they have. But here we're looking at a DNA virus, like papovaviruses. And so DNA is sort of the easiest thing for a cell to deal with, right? Because cells know how to replicate DNA, to transcribe and translate it. So when a cell gets infected with a papovavirus, remember the entire cell is engulfed and moves inside, and the capsid breaks apart, releasing the DNA. Well, our cells know what to do with DNA, right? They can easily replicate more DNA and transcribe and translate it to make viral proteins. So new viruses will assemble, and eventually destroy that host cell. But what if the virus, if its genome is RNA? That creates a bit of a problem because our cells cannot make RNA from RNA. So then how does an RNA virus replicate its RNA, right, to make more genomes, right? This poses a problem. But you just sort of know the answer to the problem, right? Since our cells cannot make RNA from RNA, there must be another way, a roundabout way, to make more RNA from RNA. And can you think of a roundabout way of doing that? Reverse transcriptase, correct. So, but our cells do not have that enzyme. So viruses with RNA genomes have to carry that enzyme with them. And so here we have an example of a retrovirus like HIV. So HIV is an RNA virus. Its genome is RNA. When it infects a cell, the problem is how does more RNA get made from the RNA? And the way is to make a DNA copy first. The problem is our cells do not have reverse transcriptase. So HIV actually carries the enzyme reverse transcriptase with it. So when HIV infects a cell, the enzyme reverse transcriptase makes a DNA copy. So okay, fine, but it's linear DNA, which a little short linear piece of DNA is going to be destroyed inside of our cells. So HIV brings another enzyme with it, integrase. And the enzyme integrase integrates the HIV viral DNA into one of the host cell's chromosomes. In this case, we call the integrated viral DNA a provirus. Remember, in bacteria, it was a prophage. But here we're talking about animal cells, we call it a provirus. So now we have proviral DNA as part of a host cell chromosome. So the DNA will be transcribed into RNA. That is the genome for the virus. And of course, the RNAs will be translated to make viral proteins. And then new viruses will be made. And in this case, they will bud, because HIV is a membrane-bound virus. So these viruses will bud from the host cell. If the host cell lives long enough, it'll produce many new particles of virus. Now, the provirus that is integrated with a host cell chromosome is permanent. It never leaves, it never excises, it never cuts itself out like a prophage, right? The provirus is permanent. So what that means is if the cell survives and reproduces, each daughter cell has a copy of that proviral DNA. So that's one of the reasons why curing HIV is not going to be easy to do, because all those cells that were infected have proviral DNA in them, and there's no way to get that out. So in order to eliminate the proviral DNA, you would have to eliminate all the cells infected with it. And all those cells are the immune cells, so we can't very well destroy all the immune cells, right? I mean, that's what HIV does, which causes the problem. So that's why the drugs today focus on stopping replication. That way the patient stays healthy. But we'll talk more about that later when we talk about HIV. But the main idea is that viruses use every possible replication strategy that exists, right, for every possible type of nucleic acid that exists. They're very diverse that way. Now, we do know that some viruses cause cancer, and they can activate what are called oncogenes inside of our cells, which cause our cells to become transformed and lose things like contact inhibition, right? They no longer are affected by density-dependent inhibition. Also, anchorage dependence stops functioning. And so the sort of interesting thing here is that we have oncogenes. Why do we have oncogenes? right? We have these genes in our cells that if they're turned on, will form cancer. Why would we want to have those kinds of genes? And the answer is, think about that fertilized egg. When the egg cell is fertilized with a sperm, you have a zygote, and that zygote then has to begin the process of mitosis and cell division, where cells grow on top of each other, forming a fetus, forming a baby, right? So at that point early in our development, we need to have genes that turn the cells on, that make them start dividing rapidly. But then at some point, those genes get turned off because we no longer want that kind of rapid mitosis and cell division. But sometimes those genes get turned back on, and that leads to cancers, to tumors. And they have found evidence in some tumors of fetal antigens. So antigens that you find on fetal cells. So there is a relationship between cancer and the fetus, right? Because they're both trying to grow rapidly. But there are some viruses that can activate these oncogenes. And so we call those viruses oncogenic viruses, right? Cancer-causing viruses. But these examples of these different virus families here, the entire family is not oncogenic. It's just there are certain viruses within each of these families that can cause cancer, but not the entire family. Otherwise, we'd all be dead now because we'd all have gotten cancer, right? The adenoviridae, common causes of colds and respiratory infections. So we've all been infected with adenoviruses. We're not going to get cancer. Herpes viridae, we're going to talk about her. Yep. Any question? So we're going to talk about herpes viridae and see how common those viruses are, but since we're all infected with multiple herpes viruses, we're not going to get cancer from them. So only certain viruses in these families cause cancer. Now, latent viral infections are analogous to lysogeny in bacteriophages. Not quite the same thing, but there are some similarities. So with a latent viral infection, you are infected with the virus, but you may be asymptomatic, so you have no symptoms. But periodically, these viruses can reemerge, and then you get symptoms, and then they go dormant again. So you may have long periods where you're asymptomatic, and then you develop symptoms, and then the virus disappears again, it goes dormant, and you have no symptoms. So this is in particular relative to the herpes viruses. So if you get cold sores or fever blisters, you know that you get them, right? You get the blisters and they heal, and then you may be good for months, maybe even years or decades before you get another one. Shingles is the re-emergence of the chickenpox virus. So the idea here is that you are infected with these viruses, but they only periodically re-emerge and produce symptoms. Now, persistent viral infections are very different. A persistent infection is one where the patient cannot bring the virus under control. So their immune system just cannot bring the virus under control. And so the infection persists for a period of time, and it is a progressive condition which is almost always fatal. So an example is subacute sclerosing panencephalitis, which is caused by the measles virus. Now, most of us get vaccinated against the measles, and even if we did not, chances are we would survive the measles, but there's a small percentage of the population that will go on to develop subacute sclerosing panencephalitis. And the problem is, it's mostly children, and almost all of them die. And that is the reason why we vaccinate against the measles. Because if your child is one of the unlucky ones that goes on to develop this disease, your child's going to die. And you don't want to be the one whose child dies from this. And so I know there are always some students who want to know more about subacute sclerosing panencephalitis, like Bryant. I know he's just dying to learn more about it, so here you go. So subacute sclerosing panencephalitis is a chronic persistent infection, and it's usually in children, or almost always in children. It can take anywhere from 2 to 10 years before you die, but it's a progressive disease, so you continually get worse and worse and worse. And only about 5% of those children or those people with subacute sclerosing panencephalitis will survive. That means 95% will die. So again, that is why we want to vaccinate against the measles. And because there are certain people choosing not to vaccinate, the United States has had measles outbreaks over the past 10 or more years, something that should not be happening at all. We should not be having any measles at all, but we are, because some people are choosing not to vaccinate, people coming from other countries who don't vaccinate against measles. So it's a problem, and it should not be a problem at all. So now we're going to talk about a bunch of different virus families. In your handouts, the families or the viruses that are underlined are the most important ones to remember. But as I said, they can be DNA or RNA viruses, either single-stranded or double-stranded. And there may or may not be a lipid membrane. It depends on the kind of virus. Our first family is the Adenoviridae family. No membranes here, but these viruses do have spikes, which they use for attachment. And they mostly just cause miscellaneous respiratory infections, colds. There are certain strains that do cause tumors in animals, but you don't really have to worry about getting any tumors from adenoviruses. But you may have to worry about adipogenicity. So that word, adipogenicity or adipogenic. So what does that word mean? Who's taken A and P? You should know what adipogenic means. All right, well, the adipo refers to adipose tissue. What about genicity? What does that mean? All right, genicity. No. Adipo, we've already established, refers to adipose tissue. And we all know what adipose tissue is, right? Fatty tissue. And so what about genicity? No. Right, forming or producing, so the creation of. And so there is some evidence that there are certain strains of adenoviruses that in certain people, if they get infected with them, it causes those people to gain weight. So this was a very interesting study that was done a while ago, where they noticed that certain patients who were overweight or obese also had been infected with a certain type of adenovirus. And so there is some evidence suggesting that certain adenoviruses, or at least one type, can cause some people to gain weight. So when it comes to potential consequences of viral infections, especially long-term viral infections, we don't always know what those consequences are, and they can be unusual things that we would never suspect. And I'll talk more about that next week when I talk about coronaviruses. In the Papoviviridae family, there's no membrane surrounding those viruses, and we're most concerned with the papillomaviruses. So human papillomaviruses, HPV. when most people hear HPV, they always think of the sexually transmitted form. But all of the human papillomaviruses are HPVs. And these are viruses that typically cause warts. And there are at least 200 types of human papillomaviruses. There are many other animal papillomaviruses, but we don't have to worry about those because, we're a different animal, so they won't bother us. But we have plenty of our own, right? 200 types. When it comes to the sexually transmitted form, if you're sexually active, at some point you will get HPV. It's almost 100% guaranteed, unless when you were young, you were vaccinated against HPV. So there is a vaccine like Gardasil, which is recommended for use in girls and boys, between the ages of 12 and 15. They want to get them vaccinated before they start becoming sexually active. And it protects against the 4 or 5 high-risk strains. So there are a certain number of high-risk strains. I forget exactly, 3, 4, 5, but they're very high risk and they cause cancer. And in particular, they cause cervical cancer in women. And so 99.9% of cervical cancers in women are caused by HPV and nothing else. So cervical cancer is basically all caused by HPV, and only those certain strains of HPV. So that's one of the driving forces for vaccination against HPV is to prevent cervical cancer in women. Now, the sexually transmitted HPVs have also been linked to oral cancers, anal cancers, prostate cancer in men. So, it is a good thing, to vaccinate, your child against HPV, especially if you have a girl, because that most likely will prevent her from ever getting cervical cancer. So the vaccine is an important one. Now, when it comes to all other HPVs, when it comes to the immune system being able to eliminate them, there is some controversy as to how effective that is or how long it takes. It's possible your immune system can eliminate the viruses and get rid of a wart, but that could take years. People with warts choose to get them frozen or burnt off, right, to very quickly destroy that tissue. So it's not entirely clear how long it takes the immune system, right, to eliminate these HPVs. But we've all got HPVs. Even if you have no visible warts, you've got HPVs because they're very common. And many HPVs do not produce any visible warts, so you never know you had them. One of the reasons why we know people have them is because there have been patients, like AIDS patients who became immunocompromised, or chemotherapy patients who became immunocompromised, and then they break out in warts. And so they were already infected. It's just their immune systems were able to keep those warts keep the virus under control. But when they became immunocompromised, no more control. So they broke out in warts. Stresses can cause you to break out in warts. There was a student once, this older student, who was into tattoos. And so he was having his calf tattooed to look like exposed calf muscle, right? So like the pictures or the models you've seen in A&P, he was having his calf tattooed to look like that. Well, that was, it's a lot of tattooing, right, to make that happen. And that was the stress, and his calf actually broke out in warts. And he had to go on an antiviral to bring them under control. And then he was going to have a lot of the tattooing redone because all the warts kind of ruined, the tattoo. So he had to have a lot of work done to fix it. But it just goes to show that we do have these viruses, right? We're periodically or frequently infected with them, but they come and go, often without causing any visible warts, but we have them. Now, I do want to take a few minutes to talk about how HPV, which infects epithelial cells, skin cells, how it maintains itself, and relate this to plasmids in bacteria, because the HPV is a circular piece of DNA like a plasmid. So we've talked about plasmids before, but there was a problem when bacteria have plasmids. So if we have a bacterial cell, I'm just going to show the cell with the plasmid in it, not the bacterial chromosome, but plasmids are extra DNA. Extra DNA requires extra energy because it has to be replicated, it has to be maintained, right? So that's extra energy. So if that plasmid does not contain beneficial genes that help that bacterial cell to survive, having a plasmid is a burden. So it's a burden to that cell. So if you had cells competing with each other, cells that have a plasmid and cells that don't, if the plasmid's not particularly beneficial, which cell type is going to win? Which cell type is going to outcompete the other one?
Without the plasmid?
Right, the one without the plasmid. So in order for plasmids to be maintained, they need to be beneficial. Well, obviously HPV DNA is not beneficial, right? So let's go back. So here we have our bacterial cell with its plasmid. It's getting ready to divide, so it will replicate the plasmid along with its chromosome, but I'm not showing the chromosome. And then there are two possible outcomes. This would be the ideal outcome. Each cell gets a plasmid, right? But of course, with bacteria, it's also very possible that you end up with this. And again, if the plasmid is not particularly beneficial, this cell is going to die out. It's going to be outcompeted, right? And you're going to end up with no plasmids. And this is a problem for scientists who try and maintain populations of bacteria with plasmids in them, especially plasmids that the scientists have created, and so they want to maintain, but they tend to get lost over time because they're a burden, right? They're not doing anything for the bacterial cells except creating a burden. Now, if we're talking about HPV, this, draw again. So this is what HPV always wants to happen. It wants every daughter cell, every skin cell, to be carrying a copy of the HPV DNA. So that's what it wants. And it has a way to guarantee that happens instead of this, right? Because if this happens, eventually HPV is going to be naturally eliminated. HPV doesn't want that. So it has evolved a very unique mechanism for ensuring that every daughter cell has a copy of the HPV DNA. And it does so by creating an attachment protein. So, oops, dang it, pen's not working. So here we see, and now my pen's not writing. So you can see the attachment protein here. And so the attachment protein attaches the HPV DNA to a chromosome in the cell, so that when all the chromosomes are replicated, the HPV DNA is replicated, and then when the chromosomes are separated during mitosis, the HPVs get separated and each daughter cell gets a copy. So every daughter cell is always infected because HPV has this attachment protein, which guarantees that the HPV DNA will never be lost. It will always end up in each daughter cell. So that's pretty amazing when you think about what HPV is doing. It's guaranteeing every daughter cell is infected, even though the HPV DNA does not integrate, it attaches to a chromosome. And not only that, but as you may recall, a lot of your DNA is turned off, right? You have all these different types of cells. So They all have the same genes, but they don't need all those genes to be turned on, right? Skin cells don't need to be making actin and myosin like a muscle cell does, so those genes are turned off. So a lot of DNA is transcriptionally inactive. Well, HPV wants its DNA to be transcribed and translated to make more viruses. So HPV not only attaches to a chromosome, it attaches to a region of the chromosome that is transcriptionally active. There are high rates of transcription there so that new viruses can be made so it can be spread. So again, a remarkable thing that this virus does with very few genes in order to guarantee it stays inside of your cells and is able to make more viruses. So, and I just wanted to relate that to plasmids in bacteria.
I have
So,
a question.
Yep.
I think we learned in anatomy that the cells in the skin, like the topmost layer, are dead cells. So does that
Yep.
affect this?
Well, the HPV is actually infected deeper in the live skin cells.
And then it
And so
comes out.
yeah. And so that's why if you've ever had a wart frozen by a doctor, they really freeze the heck out of it because they need to make sure that cold penetrates more deeply into the skin to kill all the infected cells.
I see. That's really interesting.
Yeah, and it also makes it more difficult for the immune system to get rid of.
What is the freezing? It's like a chemical, or is it?
Oh, it's if you go to the doctor, it's liquid nitrogen. And they either spray it on or they dip a swab into liquid nitrogen and then push it deeply onto the warts or to make sure that the freezing really goes deep. And it's very painful.
Wow.
But they need to make sure to try and destroy all of the infected tissue. And they also create an injury, which brings the immune system there to say, hey, we've got some injury, what's going on? And then they might now see the virus and attack it and destroy it. But warts can be stubborn, right? Because it's usually not, well, sometimes it's not just that one location where the wart is, right, that has infected cells. They may be in the surrounding tissue as well, but they're not producing warts until you freeze it, until you do damage. And often the damage can lead to a little outbreak of warts all around where you froze it because you did some damage, you weakened those cells, now the virus can multiply and create more warts. So warts can be stubborn, but not, I mean, mostly they're just visually unappealing, right? And most warts don't do any serious, cause any serious problems to you, unless it's the sexually transmitted type. Now, most of us have healthy immune systems which are able to control HPV. Yes, we may get some warts, but they're usually just a few or one, things like that. So, but there is a rare genetic mutation that a few people on this planet have, where their immune systems cannot control the wart virus at all. Not at all. I mean, the immune system works well against other things, but not against HPV. So what happens when the immune system cannot control HPV at all? And I hope you're not eating right now. Tree man. You ever heard of Tree Man or seen Tree Man? So this was an individual with that rare mutation. His immune system could not control HPV at all. And so the warts just grew like crazy. No control whatsoever. And all the warty growths looked like the bark on a tree. And that's why he was called Tree Man. When Western scientists discovered him, or when he was brought to their attention, they said, we can help. We have powerful antiviral drugs that we can give you to help not eliminate the HPV, just really, really slow it down. And then, of course, he had surgeries, right, to remove the warty growths. And you can see here where his hands are now exposed right? Because they were able to cut off all this warty growth, all this keratin and everything. But then he discovered that his hands were spongy. He had severe osteoporosis. His bones were soft because for years he hadn't been able to use his fingers or hands. And when you can't use your bones, they start to get very soft. But you can treat osteoporosis as well. Unfortunately, for whatever reason, he stopped taking the antivirals and then he died. So he died in 2016. But he had a rare genetic mutation which prevented his immune system from being able to control HPV at all. Now, students always ask, well, was it dangerous to touch him or be around him? And the answer is no. No more dangerous than anyone else, right? Because as long as you have a healthy immune system, you're mostly going to be just fine. You might end up getting a few warts, but those often happen anyway. So he was no more dangerous than anyone else would be.
I don't even understand. Where is his hands? Are they underneath?
Yep, they're all underneath. When all this was cut off, there were his fingers. But all this was underneath. All this long stuff here are the fingernails, presumably.
Wow.
Right? So his hands and fingers were under this part here.
Wow.
So, but yeah, I mean, it's really tragic. Again, he did die. So, and then I heard there was another report because he was from Indonesia, and there was another man from Indonesia with the same mutation. So, I don't know what happened to him. Moving on to the Poxviridae family. And interestingly, not all the viruses in the Poxviridae family are called poxes. Most of them are, but not all of them. So the orthopox viruses, we have vaccinia, which is cowpox. We have smallpox, also known as variola, monkeypox, which is not anything that we really worry about. Smallpox that we're going to talk a lot about. Remember, vaccinia, cowpox, was used as a vaccine against smallpox. And then molluscum contagiosum is actually a sexually transmitted infection, but it's usually self-limiting, so you don't normally even need to see a doctor. Your immune system will take care of it, and you'll be fine. But smallpox. We've all heard of smallpox, right? Smallpox is the only virus to have been eliminated from the human population. No one gets smallpox, and no one's gotten it since, I think, 1978. And that was the result of an aggressive vaccination program. And by aggressive, I mean you had no choice. Everybody all over the planet was vaccinated. And that was the only way to eliminate smallpox. So human beings no longer suffer from smallpox. It's not in the human population. It still exists on Earth in laboratories, like in the United States and in Russia and some other countries, and nobody wants to destroy it. Why do you think no one wants to destroy their smallpox stores? What do you mean by that question? If any side effects, if someone touches antivirus drugs with bare hands, probably not, but certain drugs do have side effects. So especially if you're pregnant, they have to take that into consideration. But as far as I know, there's no side effects from touching an antivirus drug. So back to smallpox. Why is it that the United States and Russia and some of these other countries refuse to destroy the smallpox they have in laboratories? Possibly for biowarfare, right? Because actually throughout human history, smallpox has been used for biowarfare, as we're going to talk about. And so there is concern that if one country destroys their smallpox, but the other one doesn't, the other one could possibly in the future use it as a bioweapon. So nobody is willing to completely destroy all the smallpox, but it no longer exists in humans. And there's a reason why we wanted to eliminate it. So smallpox does have a high mortality rate. I believe the overall mortality rate is like around 30%, but for children, it's way higher, something like 90%. So it is a very deadly disease. It killed millions and millions of people in Europe in the Middle Ages, right? So a very deadly disease. It kills a lot of people, especially if you're really young. It does cause a lot of scarring. So even if you survive, you're scarred for life. It does have a sad history in terms of germ warfare. So in more ancient times, if you had a city surrounded by walls and you were trying to attack and conquer that city, but you couldn't penetrate the walls, you could throw dead bodies over the wall that were infected with smallpox. And then the city would get infected, people would die, and the city would fall. In this country, in the 1800s, I believe, possibly earlier, but there was an incident where the U.S. Army gave blankets to Native Americans, and the blankets were infected with smallpox. And so the idea was, if your warriors are sick and dying, they can't fight. The Japanese during World War II had the most sophisticated germ warfare program. And unfortunately, they did a lot of testing on Chinese individuals when the Japanese invaded China. And then when the Japanese were forced to leave China, they left behind all kinds of nasty things. And we'll never know how many people got infected and died from the deadly germs left by the Japanese. So human beings have been using biological warfare for a very long time. And it's very, very unfortunate. But, smallpox is something that has been eliminated from humans, but we refuse to eliminate it from the planet.
If we have vaccines, is there a vaccine against smallpox?
Oh, yeah.
Right, so if we have a vaccine, even if it was, God forbid, let out, would anything even happen?
Sure. If the smallpox had been genetically modified and the old vaccine no longer works,
Oh,
right? Because we have that problem now with viruses naturally mutating. And so the vaccines we create don't work. Same with bacteria. So yes, it's possible that they could genetically modify smallpox and create a strain that is, resistant to the vaccine. And it would take time to create a new vaccine and distribute it. And by then, a lot of people would be dead.
Right. Is that the same with polio?
Polio does still exist, I believe, in the human population, but it's not considered much of a problem these days. But even people in this country through the 1950s, at least, were still getting polio. So.
Is there a vaccine against that?
Oh, yeah. I'm sure that you were vaccinated when you were a child, but so polio is still around. It's just not very common anymore because we do vaccinate a lot. So moving on to the herpes viridae family, there are 8 known human herpes viruses, HHV. So don't confuse HPV with HHV or HIV. So the HHVs, there are 8 of them, and we still use the older names like HHV 1 and 2. We still refer to them as herpes simplex 1 and herpes simplex 2. And we'll talk about genital and oral herpes more, but HHV1 is the oral form of herpes. HHV2 is the genital form of herpes. Oh, you can get them in the opposite places depending upon what you like to do, so you have to be careful. Varicella virus is the chickenpox virus. So how many of you have actually had chickenpox? Most people get vaccinated today, but is there anyone who actually got chickenpox when you were a kid? Clarice, did you?
Yeah.
Yeah, these days most children are vaccinated against chickenpox. Although chickenpox is, basically one of those harmless kinds of viruses, but one of the problems with all the herpes viruses is once you are infected, you're infected forever. But those viruses are always inside of you. But they do go dormant, but they can periodically reemerge. Now, for most people who had chickenpox, it stays dormant, forever. But in some people, it reemerges, but it reemerges as shingles. So it's a different manifestation. It doesn't look like the original chickenpox that you got when you were a kid, but you do get these patches that ooze and leak liquid, and the liquid is full of the chickenpox virus. So if you have shingles and there's somebody or in your household that never had chickenpox, never vaccinated, they can get chickenpox from your shingles. And as an adult, you don't want to get chickenpox as an adult because there are more potential complications, and also there will be scarring. Little kids, they heal up, much better, so there's very few, if any, scars from chickenpox. But when you get older, then you do get more severe scarring from chickenpox. So if you've never had chickenpox, you need to get vaccinated. And in the good old days, and you may have seen this in some TV shows or movies. I know The Simpsons did an episode once where Maggie got chickenpox. And so in the old days, in small villages and towns and neighborhoods, if one kid got chickenpox, they would actually invite all the other kids over to the house and have a pox party. And the idea was they wanted all the kids to get infected at the same time and get it over with. And that would cause minimal disruption to schools and family life and everything like that. So in the old days, they actually did have pox parties to infect all the kids at once. These days, though, we have a vaccine, so we need to do that. But that was kind of interesting that, they would do those kinds of things and say, hey, let's all get infected and get it over with at the same time. The lymphocryptovirus HHV4, also known as the Epstein-Barr virus, and this is the one that causes mononucleosis, or mono for short. And mononucleosis is very popular with college kids. Not you guys, because even if we were meeting face-to-face, you're living on campus, you're not living in dorms, you're not going to a bunch of parties where there's a lot of spit being swapped between different people, because it is transmitted through saliva. And so it does cause mono, which you will recover from it, but it is debilitating for a while. You're very fatigued. And actually, chronic fatigue syndrome in women has been linked to the Epstein-Barr virus, and so that may be the cause of chronic fatigue syndrome. And in some people, they can develop a type of cancer called Burkitt's lymphoma, but that's not very common. Cytomegalovirus, roseola virus 6 and 7, we have a slide on those. They're very common viruses. We all have these viruses. They're transmitted in saliva. We get infected when we're children, especially if you went to daycare, because anyone with little kids knows that, spit and saliva is everywhere. Little kids put everything into their mouths. And again, if you have little kids, they get sick almost every month with something, and they go to the doctor, and the doctor just says, it's viral, it's viral, it's viral. A lot of those viral are these herpes virus infections that we get when we're little kids. And Kaposi sarcoma. Kaposi sarcoma is a type of skin cancer, and you get these purplish blotches on your skin. And it was actually very rare before the AIDS epidemic. So before the 1980s, it was very rare. You would normally only see Kaposi sarcoma in people who were maybe really, really old, whose immune systems had fallen apart. Sometimes in people who were undergoing chemotherapy and their immune systems were weakened, and then the virus could reemerge and cause this type of skin cancer. But it is caused by a human herpes virus. And the virus is very common because, again, it's transmitted through saliva. So a lot of us have it. will never have any consequences from it. If you become immunocompromised, then it can reemerge and cause a Kaposi sarcoma. But all these viruses are very common. We're all actually infected with multiple herpes viruses because they are so common. They are spread through saliva. Cytomegalovirus is one. Basically, everyone, everywhere has this. The longer you live, the more likely you'll be infected. It's most commonly transmitted when we're little kids. But for most people, it will never cause any serious problems. But there are others who could potentially suffer serious consequences, especially if they become immunocompromised for some reason and this virus reemerges. And again, it was one of the problems with AIDS patients before we had all these wonderful drugs, and they developed AIDS, and they were infected with these viruses, and the viruses would reemerge, and they were nasty. The roseola viruses, HHV6 and 7, very common viruses. You notice we all have it. It's easily transmitted through saliva. We usually get it when we're little kids. It's one of those miscellaneous viral infections. Same with number 7. And again, we're not going to suffer any serious consequences from these viruses unless we become immunocompromised for some reason. Now, there is an interesting relationship between HHV6 and multiple sclerosis. So you may have heard of multiple sclerosis. It's an autoimmune disease where the immune system attacks the myelin sheath. And the myelin sheath is what surrounds and insulates nerve cells. And it allows for efficient nerve impulse conduction. But when the myelin sheaths are damaged, those nerve impulses are not conducted well. And this leads to a variety of neurological symptoms. So there is some evidence suggesting that some people who may be predisposed to developing multiple sclerosis, it's triggered by infection with HHV6. But what are you going to do? We've all got it. It's very easily spread. It's impossible to avoid it. And there's no vaccines for these. So, you're kind of stuck. Now, I've talked a lot about chickenpox, right? When it gets reactivated, it manifests as shingles, but can cause chickenpox in people that have never had chickenpox or never been vaccinated. With the simplex virus, oral and genital, as I said, you can get them in the opposite places, so be careful. And once you're infected with these, you are infected for life, right? They never go away, but they do go dormant. And so you have long periods of time when you're asymptomatic. But then you may get an outbreak, right? You may get cold sores, fever blisters, or genital blisters. Usually, the longer you live with this, the fewer outbreaks you get. But when simplex viruses reproduce in your epithelial cells, they destroy those epithelial cells. That's what causes the blister. Now, when it comes to, and then these herpes viruses, what they do, they go and hide inside your nerve cells. And the immune system cannot get to them inside your nerve cells because the immune system is not going to attack and destroy your nerve cells. That would cause a lot of damage. But this is where herpes viruses hide, in your nerve cells. And periodically they reemerge. And then they go run and hide again, right? Because your immune system is fighting them, so they have to retreat and go hide inside of your nerve cells. And that's why we can't get rid of them. When it comes to something like oral herpes, cold sores, and fever blisters, stress can cause an outbreak. Sunburn on your lips can cause an outbreak. Colds, fevers, so any kind of immune stress can cause an outbreak. Menstruation can cause an outbreak of genital herpes. So basically, stresses to your body can lead to outbreaks of these herpes viruses. The problem, especially with genital herpes, is that you need not have a blister to be shedding the virus. So you can be in the virus and have no other symptoms. Now, when it comes to the number of infections, this 45 million is an old number. It's much higher than that now. And in particular for women in the United States, depending upon where you are, up to 50% of women are infected with genital herpes, and most of them do not know it. And the reason why they don't know it is because the blisters can be inside the vagina where they can't see them or feel them. On men, it's much more obvious, right? We have a blister on the penis, you can see that. But for women, it's not always that obvious, which is why a lot of women do not know they have it. And if you don't know you have something, you spread it because you don't take any particular precautions or protection against that. And so that's one of the reasons why the herpes infection rate is so high. And this is this YouTube video is actually comic relief at this point. I'm not going to show it, but you can watch it on your own. It's not about herpes, but it's about chlamydia, but it's a funny song if you want to go back and listen to it. So we're going to stop there for today. Any questions or comments? Okay, then we'll start lab at about 10 after. All right. So I'll see you guys in lab.